Take a look at the diagram on this slide. This is a diagram that you'll find in lots of different textbooks. It is a picture that represents this really important concept called energetic coupling. Remember, energetic coupling is simply the way that the ATP molecule is used in a living cell. It shows us how a cell couples exergonic reactions, those are the catabolic type, with endergonic reactions, those are the anabolic type. And it does that coupling with the intermediate sort of middleman molecule called ATP. So let's walk through the diagram together. In the center of the diagram, you can see that chemical reaction that we've been looking at. We can take ADP, adenosine diphosphate, and an inorganic phosphate, or a phosphate group, and we can put them together with a little bit of energy, and we can create ATP. Now, a little bit of water is also created during this reaction, but don't worry about that. The ATP molecule can then be broken apart, and that's what this arrow is showing us, to produce ADP plus phosphate plus a little bit of energy. Now, what the cell does by breaking and then building and then breaking and then building ATP is it couples reactions that are going on all around the cell at any one moment. On this side, we're going to think about all of the catabolic metabolism that goes on in a cell. All of the exergonic things that are happening. All of the things where bonds are being broken in molecules. That's catabolism. Every time bonds are broken, electrons are released. Energy is released. If the cell didn't do something to harvest that energy at that moment, some of that energy will be lost as heat to the universe. The cell wants to grab onto that energy right now and hold on to it so it doesn't get lost. But if there's no anabolic reaction going on in that second, how else can it grab onto the energy? Well, it can grab onto it right here. It can use the energy to put a phosphate group onto ADP and make ATP. It has harvested that energy over here. It has harvested the energy by storing it in this molecule. Then in other parts of the cell, on this side of the diagram, where anabolic reactions happen to be occurring, where molecules are being built, energy is needed to do that, to make a covalent bond. You need some energy. How are we going to get that energy? Well, we're just going to break that ATP. We're going to take the phosphate group off, making a DP, and that little bundle of energy will be released. And it can be used to fuel these processes. This is energetic coupling. This is how a microbial cell, or any cell, uses ATP. Again, it can be confusing for students because half of the coupling diagram shows ATP being made, right? Half of the coupling diagram is this reaction. ADP and a phosphate group and a little bit of energy becoming ATP. Sure looks like ATP is being made, but I don't want you to think about coupling as the way a cell produces ATP, because that's happening in a completely different set of reactions. That is what is happening in respiration. So cells use ATP this way. Cells build ATP, produce ATP in other metabolic reactions. Let's spend a minute and just introduce those. In eukaryotic cells, like human cells, there are mitochondria that are devoted to the production of ATP in a process that we call cellular respiration and oxidative phosphorylation. Now, you may or may not have heard of these terms before, but we're going to start talking about them in detail. Cellular respiration and oxidative phosphorylation are two processes that go together to produce ATP. When we talk about cellular respiration, we're really talking about a cell taking glucose and breaking it apart, breaking it down all the way to a one carbon molecule called carbon dioxide. During that process of breaking all those bonds in a glucose molecule, a whole lot of electrons are going to be released from those bonds, and they need to go somewhere. The electrons have to be received, and we talked about how aerobic organisms will use oxygen ultimately to receive those electrons. But then there's a second part of the process. The energy of those electrons will also be used. The energy that those electrons carry with them, because of course they are the conduits of energy. That electron energy will be used in a process called oxidative phosphorylation to build ATP. Now here's something I want us to know, and you'll hear me say this again and again over the next couple of lectures. This word oxidative does not mean oxygen. Let me say that again. That's very important. Oxidative does not mean oxygen. Oxidative refers to a type of chemical reaction called a redox reaction. We're going to talk about redox reaction in the second lecture of metabolism. But for now, understand that this has nothing to do with oxygen. This has to do with the nature of the chemistry that goes on in this series of reactions. This series of reactions that is ultimately going to produce ATP for the cell. So, eukaryotic organisms build or produce their ATP in the mitochondria. That's what the mitochondria's job is. Prokaryotic cells don't have mitochondria. So where in the world do they produce their ATP? Well, they do it across their cell membrane. It turns out that the inner mitochondrial membrane is key to the production of ATP. So it's the inside membrane, that inner membrane, that is the critical structure for the production of ATP inside the mitochondria. Prokaryotic cells don't have mitochondria, and they need some membrane to make ATP, so they just use their cell membrane to do it. What makes microbes so fascinating is that they can make ATP using their cell membrane in a couple of different ways, very different from human cells. Prokaryotic cells, bacteria and archaea, can do aerobic cellular respiration. In other words, they can do this in the presence of oxygen, and they use actually two processes to make ATP this way. They use oxidative phosphorylation again, and they use a process called substrate level phosphorylation. If you've never heard of that before, don't worry. We will talk through it. Prokaryotic cells, some of them at least, are also capable of doing a process called anaerobic cellular respiration. In other words, cellular respiration in the absence of oxygen. And they also use oxidative phosphorylation and substrate level phosphorylation to make the ATP. Now, I'm just going to mention this last point. We're not going to talk about it in any detail. There is a third way that microbial cells make ATP, and that's called photophosphorylation. Don't forget, some of our microbes are phototrophs. They are capable of harvesting energy from light, and they can use that to make ATP through a process called photophosphorylation during photosynthesis. Don't forget about our phototrophic microbes. So, there are a lot of terms involved in the production of ATP. There's a lot of processes that we have to wrap our heads around and learn the details of. And you may have only ever referred to this as respiration, as cellular respiration. But as I said a minute ago, cellular respiration is technically only part of the process. Cellular respiration is added on with oxidative phosphorylation or substrate level phosphorylation to actually build ATP. So we need to understand these two processes. Oxidative phosphorylation turns out to be a very good way to make ATP. And again, this goes on in all of your cells in your mitochondria. Oxidative phosphorylation also goes on in a lot of microbial cells. It's very, very efficient. In other words, for each glucose molecule that enters into cellular respiration, a whole lot of ATP gets made. Now, ATP that's produced through oxidative phosphorylation is going to be produced using something called a proton motive force in those redox type of reactions. And as I said a minute ago, this can be either aerobic or anaerobic, depending on the microbe. Some microbes are going to use oxygen as the ultimate or final electron accepting molecule. Other microbes are going to use things like nitrate or sulfate or carbonate as their final electron acceptor molecule. Aerobic and anaerobic oxidative phosphorylation. We're going to leave this whole big complicated bit of information for today. We'll talk about that next time. But let's finish up today by talking about this second mechanism called substrate level phosphorylation. This is a way for a cell to make ATP. It just happens to be quite inefficient. In other words, for each molecule of glucose that gets broken down in a cell, only a small amount of ATP can be made when you use substrate level phosphorylation. The key to this process is that word substrate level. You see, the ATP is going to get made in sort of a piggyback fashion as an enzyme is catalyzing another reaction. a different enzyme is going to be doing its business, catalyzing a chemical reaction somewhere in the cell, and the cell is going to piggyback a little bit of ATP production on top of that. Let me show you what I mean. On this slide, the green sort of globular thing represents an enzyme. And there are several enzymes that can be used for substrate level phosphorylation in living cells. So this is just some enzyme. And as usual, the enzyme is large, it's three-dimensional, and it has some area on it that serves as an active site. That's what these two little divots here represent, active sites. Now, it's hard to see, but you can just make out there's a little structure in here in white. This represents some substrate molecule. And it happens to have a yellow circle drawn on it that represents a phosphate group. So you could think of this as being a phosphorylated substrate. Some substrate that fits perfectly in this active site. It's going to become some kind of product molecule. This is just the way enzymatic reactions occur, right? Now, look up in the second active site. What fits perfectly in the second active site is ADP. So this is what I mean when I say substrate level phosphorylation piggybacks on top of an enzymatic reaction. There are two very different active sites in this enzyme. One for its ordinary substrate and one for the ADP molecule. Now, what's going to happen during the chemical reaction? Remember, the enzyme is going to bind with the substrate. It's going to change shape. The old bonds will be broken down. New bonds will be made. Product will be produced. And that product will be released from the active site. That's what they're showing us here. The old bond that got broken down was the bond holding this yellow phosphate group on. So now the white molecule, that's our product. And that little yellow circle, that phosphate group that got released, that's going to be added on to the ADP to make ATP. And the ATP will be released. So it's a one-to-one reaction here. For every molecule of product that this enzyme creates, it also makes one molecule of ATP. Very inefficient. This is a very inefficient way to make ATP.